So hi everyone, I'd like to welcome everyone to a special interview that we have with Dr. Jessica Lin, who took over from Dr. Alice Shaw at Massachusetts General Hospital, which we'll refer to as MGH from here on. And we have Dr. Justin Gainor. Dr. Gainor was one of the awardees for our 2018 LUNGevity Summit, no, not Summit, but LUNGevity Awards, and it was a 2-year project. The project would have been getting very close to finished at this stage, but with COVID got delayed a little bit, but I'm sure Dr. Gainor is expecting to be finished before the end of the year, and so we're really honoured to have them back with us again. I want to thank them so much for preparing for the summit and presenting at the summit. Dr. Lin was just telling me that she watched pretty much the whole day, and so we're really honoured to have them with us. I'm really apologetic to both Dr. Gainor and Dr. Lin for just simply not having enough time for them to be able to do the presentation that we would have liked to hear, but that's why we're back here again. And very grateful to both of you for taking time out. So I'll let you both do a bit of a self-introduction about how you got into being in the ALK positive world, and how you got into oncology for Dr. Lin, and how you got into research for Dr. Gainor. And after you've done a bit of an introduction, you can go ahead with the presentations that you're going to do the other day, but in a much less rushed manner. And then I'll have some questions for you both during the presentation and afterwards. So I'll hand it over to you. So thanks again.
So I can start. Hi everyone, we're so excited to be here and to reconnect with all of you through this forum. Like Colin was saying, I really watched almost all of the summit. I was there and it was just such a wonderful experience. I guess in terms of how I became interested in oncology, I first became interested actually way back in my undergraduate years when I majored in biochemistry and happened to meet a mentor who was an oncologist, and his passion for research and how he cared for patients is what really got me interested. And then going forward in the medical school, residency, and fellowship, for some reason, I found myself really connecting in a special way with lung cancer patients in ways that were different from others. So I realized that that was where my passion was. And then importantly, I also met mentors along the way that kind of sealed the path for me. That includes Dr. Alice Shaw, Dr. Gainor, others in our group who really have unparalleled way of dedicating themselves to our patients and letting that drive the research. And so I decided that's what I want to do, and that's why I'm here.
Terrific, thank you.
Yeah, I also wanted to thank you, Colin, for arranging this. This is a nice, I think, opportunity, especially in these trying times with COVID. I think we all want to find ways where we connect, and finding novel ways to do that, you know, using technology so we can all kind of be together as a community and share insights and you know, answer questions. So we're really grateful to have been a part of the summit, and really grateful for this opportunity today. Like Dr. Lin, what drove me to oncology was also people, and in some ways it's you know, my career has and we often talk about serendipity in medicine, and I think that certainly was the case in my my career. You know, when I entered medical school, for reasons beyond understanding now, I thought I wanted to be a surgeon, which I think would probably make Jess laugh. Probably what, you know, knowing what I know now, but you know, I had applied, you know, early on. I recognized the importance of research and driving discovery and helping patients. And so, you know, while I was thinking I wanted to be a surgeon, I had applied for a research after my first year of medical school, a research fellowship, and 6 people got assigned to surgeons, and 1 person got assigned to an oncologist, and I was the one assigned to an oncologist. And that really, I think, changed my whole way of thinking of how important it is to develop longitudinal relationships, great relationships with patients, being there through one of the most trying times of their lives, and it was the perfect balance of kind of the humanism of patient-doctor relationship as well as the science and that fusion. And then when I became a resident, also serendipity stepped in where I thought I was going to go into a different area of oncology. I thought I wanted to be a GI oncologist. And for whatever reasons, people weren't getting back to me, and I finally reached out to someone in our group, and they said, there's this fabulous new thoracic oncologist. This was 2008, who just came on staff, and her name is Alice Shaw, and she's working on this new gene that just got discovered in lung cancer, and it's called ALK. And really, since that time, that's when I first started working with Dr. Shaw when I was an intern, and I would say the rest is history in that kind of really. And I think one thing that motivates both myself and Dr. Lin is I think the not doing research just for research sake. I think it's asking questions that at the end of the day, it's not about writing a paper or anything. Is this question going to help people? And really framing everything that we're asking in that through that prism of is this something that we can directly tie into the clinic and help people? And I think that kind of underlies the projects that you're going to hear about today.
So if you had to look back over all the research that you've done in all those years since, and I'm sure you've published many papers, and probably in the hundreds, I'm going to guess. If you had to look back, what research project have you done that you feel has had the biggest impact on patients' health and lives? If you had to go to one paper.
Yeah, if I had to say one paper, I'm going to maybe split it and say 2, but
Okay.
But they're related. I would say one is in 2016 we published a paper that was the first paper really describing the whole spectrum of resistance to ALK inhibitors, not just crizotinib, but it was the first paper showing that all the different second generation inhibitors, alectinib, rechallenging, crizotinib, how resistance changed from crizotinib to these second generation inhibitors, and also showing that then lorlatinib, at least in those preclinical tests that we did, was able to then overcome those mutations. So I think that is the paper that I'm most proud of. It was years of work, but it was, because it's also, at the end of the day, I think it's helped inform, studies like the ALK Master Protocol that Dr. Lin is leading, really showing the importance of understanding resistance to targeted therapies and not just defining it, but then how do we intervene once we know what's there. So I would say that's the paper I'm most proud of.
Very good.
Yeah.
Just out of interest, we just conducted a little mini survey of our group. We had about 370 responses within 24 hours, which is pretty phenomenal when you stop and think about it. And the first question was asking which ALK TKI they're currently taking. And there's a few that are not on any TKI, of course, but the vast majority are on a TKI. And the percentage now that are on lorlatinib is up to 15%, and if you really stop and look at all the ALK TKIs, you might start off on different roads, but everyone ends up at the lorlatinib road. So ultimately, out of all the TKIs, there's probably only one TKI that we're probably all going to land on, and that's lorlatinib. You know, people might start on brigatinib, or they might start on alectinib, or even ceritinib, but you know, ultimately they end up, unfortunately, sooner or later, probably going to end up on the lorlatinib pathway until we come up with something totally new and different and better. So yeah, it was pretty interesting. So specifically, is your research now more focused on immunotherapy, Dr. Gainor?
Yeah, you're right that much of my background was all in targeted therapies, and beginning around 2015 or so, we started seeing this early data on immunotherapy, and so over the last several years, I've tried to bridge the gap between the targeted therapy world and the immunotherapy world, and really trying to understand how do we use immunotherapies in patients who have these oncogene defined cancers, things like EGFR or ALK positive cancers, and trying to understand what the immune system is like in those tumors. And so that's how I've tried to marry my interests of targeted therapy and immunotherapy together. And I think that's at the heart of this grant.
Terrific. So back in 2018, LUNGevity put out what's called a request for applications, which basically is where you can explain it better than I can, but it's where we sought to receive applications for research that we wanted to fund. And you were one of the applications. We had 14 applications in 2018. And tell us how you found out about that award. How do you even find out about what grants are available?
Yeah, it's a good question, because I think it speaks to kind of your efforts as a community. We have now a team meeting once a month, where part of that is letting one another know about grant opportunities. And so we have a grants manager. Sometimes we hear about things that way. Other times we hear about it through social media, various specialty organizations, things like that, emails. So I had heard about it. I do follow the LUNGevity Foundation. I've been very aware of everything that the ALK Positive community has done, and through those resources, I heard about it through actually multiple different people, and knew that it was a mechanism that we could make a substantial contribution to. And so, for those of you who aren't familiar with the path, it started with just like an initial letter of intent, just what a broad strokes of what we were interested in terms of questions, and then based upon that being successful, a much more detailed plan for how we were going to pursue the questions that we put together.
And that's a pretty time-consuming process, isn't it, Dr. Gainor? How many hours would you say goes into doing a grant proposal?
It's Dr. Lin knows my writing style of it's writing and rewriting and constantly. I think I hate everything I write the first 10 times, and so I think it's just constantly trying to perfect in your ideas and getting feedback from other people, and so that's why we're presenting this as a team, but it's more than just the two of us too. It's constantly getting feedback and feedback from people who are also outside of your prism. Is I think at the core of this is also a multidisciplinary approach, with basic scientists, immunologists, people who can add different perspectives. I think that's really important. And so having
a meeting this morning with Dr. Basu Roy at LUNGevity, because we're getting towards the end of the selection process for our 2020 awards, which is substantially higher budget than what we had in 2018. And one of the things that Dr. Basu Roy pointed out was that what's quite unique about the mechanism that we're doing compared with when you do, say, other grant applications, is that we actually provide feedback and ask you more questions to allow you to further refine your proposal. And it does require more work on the part of the applicants, but at the same time, I think the final proposal ends up being a lot more on target and a lot more focused. So it's a really
and I think it also meets, it allows all sides to kind of meet in the middle and prioritize, what's most important, what's most impactful,
right.
And coming at an idea that way. Exactly. And it's, I think, as all good science is, it's idea, test the idea, and then refine your idea, test again, and the same thing goes just when you're putting ideas on paper. Putting them down, coming back to them, asking other people to weigh in, and just trying to perfect it.
So I'll know that everyone's main thing is they really want to hear about the research that you've been doing over the last 2 years, and what were you trying to find, what was the hypothesis that you had, what were the aims that you had, and very often with research, unfortunately, that they do the research and their hypothesis ends up not being anything. But in this particular case, it seems like your hypothesis is getting validated, which is absolutely great news for all of us. And so we'll let you and Dr. Lin talk about the research that you have been doing, and as much as you're able to disclose what you found out so far, please do so. And I know basically immunotherapy hasn't been working for ALK in the vast majority of cases. On the other hand, the risk of it actually seems to quite often cause hyper-progression, so it's something that at this stage is being avoided. But on the other hand, we know that immunotherapy can be immensely effective, and it can actually work to the point where you don't even have to keep on taking treatments. You can actually, have the body doing its own immune defense system. So we're going to let you talk about your research and how you are looking into turning what they call a cold tumor, which means a tumor that doesn't respond to immunotherapy, and trying to make it hot, which means that it will respond. So I'll hand it over to both of you.
Great. Thanks so much.
Yeah, so I can start
let me make sure I've got screen share set up here for you. Hang on one moment.
There you go, it's working.
Okay, great.
You should be able to see my slides. So our project was entitled Overcoming Innate Immune Resistance in ALK Rearranged Lung Cancer. This is also work in collaboration in addition with Dr. Greg Riley at Memorial Sloan Kettering as well. So as most of you in the audience know, our treatment paradigm in ALK positive lung cancer was really transformed by the availability of targeted therapies with ALK inhibitors. And so just a little bit of history, ALK rearrangements were first discovered in non-small cell lung cancer back in 2007. And then we have had multiple distinct ALK inhibitors become developed since then over the past decade. And so crizotinib was the first ALK TKI to be approved in ALK positive lung cancer. This was back in 2011. This is what we call the first generation ALK inhibitor. It also has activity against MET and ROS1 as well. And then came the second generation ALK inhibitors. These include ceritinib, alectinib, and brigatinib, which initially were developed for patients that had previously received crizotinib and either had trouble tolerating it or progressed on it. But then more recently, ceritinib and alectinib earned the approval for the first line indication for patients who are newly diagnosed. And then in 2018, we saw the accelerated approval of lorlatinib, which is now what we call third generation ALK inhibitor. It's very potent, including in the brain. And this is right now approved for patients who've received ceritinib or alectinib as the initial therapy, or received crizotinib followed by another ALK inhibitor. And so here we're often able to treat patients with ALK positive lung cancer with one ALK inhibitor after another in a sequential manner. So at this time, the standard initial therapy for ALK positive lung cancer is alectinib. And then after the disease progresses on alectinib, we have some options. This includes lorlatinib, or in certain instances, we may explore another second generation ALK inhibitor like brigatinib. Of course, we face a challenge here, which is that in most patients, resistance inevitably develops to these ALK inhibitors, emphasizing that we need to identify additional treatment strategies, ideally other than targeted therapies as well, that we can use to treat advanced ALK positive lung cancer. And so on the left here, I've inserted two pie charts. These pie charts are looking at different ALK resistance mutations that are detected in the tumor biopsies that are taken from patients progressing on a second-gen TKI. This includes alectinib or brigatinib, as I mentioned, or those taken from after disease progression on lorlatinib. So this was a work building off of Dr. Gainor's initial publication that he mentioned, the work that he's very proud of from 2016. And then we continued to collect biopsies from patients for research and clinical care as well. This is from our center at Mass General. And what I wanted to highlight here is that what you can see after lorlatinib is the emergence of these complex double and sometimes even triple ALK resistance mutations, which becomes very challenging to treat. And then in over half of the cases, we're not seeing any ALK resistance mutations, which suggests that there may be another important bypass pathway that is allowing the cancer cells to continue to grow. That's another big challenge that we face right now in clinic. So that's where we really hope to explore the utility for immune-based therapies. And so in lung cancer, we have referred to targeted therapy as a real breakthrough in our treatment. And immunotherapy became another major breakthrough, not just in lung cancer, but in other solid tumors as well. The reason being that we've seen real benefit from immunotherapy in our patients, in some cases, as Colin mentioned, with durable benefit as well. And so we have had a number of immune checkpoint inhibitors become FDA approved in lung cancer. These are drugs like pembrolizumab or nivolumab. There are a number of others as well. But the issue is that not all patients derive benefit. And as a matter of fact, as mentioned earlier, our ALK positive lung cancer patients, we don't tend to see major responses from these checkpoint inhibitor therapies. So this is work that was published by Dr. Gainor and his colleagues back in 2016, where he specifically looked at the efficacy of these checkpoint inhibitors in patients with lung cancer that have either ALK rearrangements or EGFR mutations. And so here you can see that the rate of responses to these agents were significantly lower among the EGFR mutant and ALK positive patients compared to those with wild type lung cancer. And on the right, you can
explain for the benefit of our members what you mean by wild type.
So these are, on the right side of the bar graph, are the patients that have lung cancer that are not found to have EGFR mutations or ALK rearrangements. So on sequencing test, it would have shown no EGFR mutations, and ALK testing looking for rearrangement would be negative.
So wild type means that you don't have that, is that correct?
That's right, that's correct.
Good.
And then on the right side is the progression-free survival curve. So progression-free survival is the length of time between initiation of therapy and the progression of the cancer or clinical deterioration. And the dark curve here is looking at the patients with the EGFR mutations or ALK rearrangements. The dotted curve here is the patients with wild type, the no EGFR mutation or no ALK rearrangement. And what this is showing is that the progression-free survival on these checkpoint inhibitors was much shorter for patients with ALK positive or EGFR mutant lung cancer. This is one example of several studies that demonstrated the limited activity of immune checkpoint inhibitors in these oncogene-driven tumors like EGFR mutant lung cancer or ALK positive lung cancer. And so that was the basis of the key question that really led to Dr. Gainor's proposal, which is how can we take the transformative benefit that we've seen with immune-based therapies in some patients with lung cancer and try to bring it now to our ALK positive lung cancer patients. That is the main question that we're trying to address here. And we hypothesized that for ALK positive lung cancer, distinct immune-based strategies may be required. And so in order to rationally inform the development of these distinct immune-based strategies, a multi-pronged approach was proposed. So these are the three specific aims. The first aim, for example, is looking at factors that may be intrinsic to or inherent to the tumor cells themselves that may be important for immunogenicity of the ALK positive cancer. Also looking at the intersection between the cancer cells and their surrounding microenvironment, which includes the immune cells that may be infiltrating that environment. And then also looking at T cell responses in the bloodstream that may be specific for ALK. In thinking about this study though, one challenge that was foreseen was that while ALK positive lung cancers affect many patients, it remains a relatively small subset when you look at the non-small cell lung cancer as a whole, right? It only comprises 3 to 5% in general of lung cancer. And so to overcome that challenge, we have developed this strategy of partnering with the ALK positive community to establish an infrastructure that facilitates a larger scale acquisition of specimens and clinical data that is not limited geographically. And so this was in collaboration with the Adario Lung Cancer Medical Institute, also called ALKME, where we developed a protocol that would basically allow us to develop a web-based, patient-driven IRB compliant platform to obtain informed consent from patients across the nation that would then allow us to gather the clinical data and the tumor specimens that may be needed for genomic analysis and immune profiling.
Will this be only eligible for people in the USA?
So this is for patients across the states in the United States.
Okay, not for Canada or anywhere else.
Right now, it is not, and it has to do with the regulatory requirements, the legal restrictions that bar us from practicing medicine or doing research across boundaries. This is something though that we recognize that lung cancer has no boundaries, and so it is certainly something that we're going to work towards. How do we either partner with international researchers so that we can eliminate those barriers while also pursuing additional regulatory input from our governing bodies. So, but it's a great question.
Yeah, it was one of the questions one of our members asked, so thank you for answering.
Yeah, so this protocol is actually all written up. We have entitled it ENIGMA+. It's actually short for Elucidating Novel Immune and Genomic Markers for ALK. And we hope that this will be fully IRB approved and ready to go in the next 2 to 3 months. So we wanted to let all of you in the community know about this study so that we can get a lot of interest and gather the data that we need to achieve our goals. And so what are some of the questions that we could explore with these tumor specimens? So one example is looking at, as I mentioned, the tumor intrinsic factors associated with immunity. And so once we have the tumor specimens, we could do sequencing and look at what we call tumor mutational burden, which is the overall amount of mutations across the genes in a tumor. We know that can correlate with the burden of what we call neoantigens, which are foreign antigens that the immune system can then recognize, and it can then lead to triggering of an immune response. And we have some preliminary data shown here that suggests that ALK-positive lung cancers may have a lower overall tumor mutational burden. It may be overall a quieter genome compared to other lung cancers, but we really hope to verify this at a larger scale using these tumor specimens from additional patients.
And again, just to clarify for our members, there's quite a distinction, quite a difference between tumor mutational burden and tumor burden. And if you'd like to just clarify that difference, because I think to a lot of people that might be fairly confusing.
Yeah, I can, so you're right. So tumor burden, I think what you're getting at is, if you look at a CT scan, how many, areas of cancer we see in the body, and which also reflects, and gives input on symptoms that people may be having. Here we're looking at something very different. We're looking at basically how many mutations there are within a cancer specimen. Mutations are just a fancy way of saying misspellings in the DNA. And it is a bit intuitive here that basically the more mutations, the more misspellings in a cancer, the more foreign it may look to your immune system, right? The way our human bodies are able to recognize bacteria, viruses, because they look different. The problem here is that cancer cells derive from us. They were once normal parts of ourselves. And so how close are they to that normal versus how far away? And that's essentially what we're talking about when we're saying tumor mutation burden.
That's a really good explanation. Thank you.
Okay. And then another question we can explore is what we call immunophenotype in ALK-positive lung cancer. So this includes looking at things like PD-L1, which is a known biomarker that tends to correlate with benefit from checkpoint inhibitors like nivolumab and other ones that are approved in lung cancer. Now, in ALK-positive lung cancer, based on our preliminary data, there are certain tumors with ALK rearrangements that have high level of this PD-L1 marker, which in other subsets of lung cancer, we might use to say, okay, well, this tumor may respond well to checkpoint inhibitors. However, what we found in our experience is that PD-L1 expression in ALK lung cancer specifically doesn't tend to correlate as much with benefit from immune therapies. And this gets back, Colin, to your remark on ALK lung cancer being cold tumors, where even this high level of PD-L1 may actually be intrinsic upregulation because of the oncogene ALK rearrangement, and not really having to do with activation of the immune system or infiltration of the immune cells, which tends to be more lacking in ALK-positive lung cancer.
This is a critical point, and if you wanted to ask us about kind of in our initial observations, what is the most, I think, impactful for patient care, I actually think this is one of the most immediately relevant to ALK-positive patients, because essentially what it's saying is that this PD-L1 test that every oncologist is sending can be a red herring if you're ALK positive. And it can redirect people to the absolutely wrong therapy. It can redirect them away from an ALK treatment and towards immunotherapy, which is the wrong initial approach. And so one of the key things that we want to do is emphasize to oncologists, that this is not a reliable test. Even if it's high, this is not, don't just jump. It's not like ALK itself. It has different meaning here.
Thanks for bringing that topic up. And if I can just take you away from your presentation just for a minute or so, but we don't see it too often, but sadly we do get members of our group that join the group and their doctor had put them on immunotherapy, even though they have had testing that showed that they were ALK positive. And I know you just mentioned that you're trying to get the word out to the rest of the oncology world. How do you go about getting the word out? How do we get that consistency of standard of care?
Yeah, maybe I'll give my thoughts, and Dr. Lin, please jump in. I think one of the challenges is that the timing of the testing differs. So the PD-L1 test, oncologists will get back in a day or two, but for ALK testing, sometimes it can take 2 to 3 weeks. And there's always that push of, I want to get started on treatment quickly, and we all have that. The challenge, though, is that here it's taking longer to get the genetic results, and I think that leads to that tension. And so for us, part of it is education, continuing to we highlight cases, because it's not just that it's if you start with an immunotherapy first and then do an ALK inhibitor, it's not that you've started the wrong treatment first. It's that you can then see more side effects with the ALK inhibitor if you start it after immunotherapy. And so, this is always a scenario that when I'm asked for cases to highlight,
So, Dr. Gainor, your sound went really. What happened?
All right, is this better?
Much.
Okay. This is a situation that when we're asked to present cases, at educational conferences, it's situations like this that I like to highlight. A situation of someone who their genetic results aren't back yet, but their PD-L1 is high. The answer is, unless someone has significant symptoms that really require starting treatment, we should be waiting for the ALK testing and all the other genetic results.
And would you suggest, if someone is in a situation where they've just got to start doing something really soon, would you suggest that they start maybe just on chemotherapy only?
Absolutely. That's what, so if the gene testing is pending and they need to start therapy right away because of the disease burden and symptoms, that's absolutely what we would do, is start with chemotherapy first, get a dose in. And then once the gene testing comes back, if there is a targetable oncogene like an ALK rearrangement, then we can pivot and switch to targeted therapy. That's what we would recommend. And holding off on the immunotherapy until we get the full molecular testing back.
I'm going to tell my personal story. Back in 2016 when I was diagnosed, and I wasn't doing terribly well when I was first diagnosed. I was in a lot of pain, had fairly substantial tumour in my lower spine, and my doctor wanted to start me on chemotherapy, just as you suggested, Dr. Lin, and me being just knowing just enough to be dangerous, did some research and didn't like the idea of doing chemotherapy, and I didn't know anything about ALK. I didn't even know that she had done biomarker testing for it. She had, for which I'm very grateful, but I was not aware of that. And she wanted to start me on chemotherapy, and I did my research, and it said, well, if you do, statistically, and as we know, statistics are just a generality for everyone and doesn't point to the individual responses, but the overall theme I got was that, without chemotherapy I might live, 6 to 12 months, and with chemotherapy I might live an extra 3 to 6 months or something like that. And for my point in life, I was more interested in quality of life than quantity of life. And on the other hand, I'd read that immunotherapy had a chance of providing a lasting response, and I was like, I'd rather take that chance to get a lasting response. And so I actually did start on Opdivo, and then I got my result back, and it came back ALK positive, even though I'm actually squamous, so I think it was a little unexpected. And then I started on crizotinib, and the interaction between Opdivo and crizotinib is now known to be really, 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 really toxic. And so I got switched out really quickly to alectinib, which probably was a blessing in disguise. But that was my story. It was me, the patient, pushing for the immunotherapy, not the doctor. So
No, and I think that is one of the allures of immunotherapy, and I think at our core here is maybe it's not just maybe it's not PD-1 that is the right lever that we need to press on the immune system. What Dr. Lin has highlighted on this slide is, one of the other things we're doing is we're not just looking at PD-1 or PD-L1 here, is now looking at all of these other brakes on the immune system. We like to think about the immune system like a car, and then it's got gas pedals and brakes, but it's far more complicated. It's got multiple gas pedals and multiple brakes. And so I'll hand it over to her, and she can just show you on this slide. That's part of the core of this grant is looking at multiple different gas pedals and brakes, and are there other levers that we need to be pressing for ALK?
Yeah, and so what you're looking at on the right side here is different, beautiful colors. Each color looking at really the expression of a different marker that we think is relevant to immune response and immunogenicity. And our team in pathology has developed this multiplex immunofluorescence assay that allows us to use minimal tissue, because tissue is so precious in trying to think about how to allocate what amount to what specific assay. So this allows us to use very small amount of tissue to look at the expression of various different markers simultaneously. And we think this is going to be a really important tool in evaluating the different tumor specimens that we're able to gather through the ENIGMA protocol. And then the last aim was looking at the anti-ALK T-cell responses. So why we want to look at this has to do with immune-based therapy that is called cellular therapy. So cellular therapy, the idea here is that you might be able to harvest or collect T cells from the blood of patients, and then the hope is to modify or engineer the T cells in a way that may boost the likelihood of triggering of an immune response against, in this case, ALK. And so this research is ongoing, where we're collecting blood samples from our patients and looking to see if we can detect native endogenous T-cell responses in our patients with ALK lung cancer, because that will be the first step in trying to achieve and enable these types of cellular therapies. And ultimately, our hope is to not just gather and publish these data, but really to be able to share the data and the lessons that we learn with the broader ALK patient and research community. And so this, I put it up here as just an example of how that may be enabled. This is from a CBio portal, which is developed actually by MSK. And you can see how it allows you to visualize very large amounts of data. This is for breast cancer, but it's looking at clinical outcomes, the overall survival. It's looking at the genome landscape and the mutation counts, protein expression. And so this could be one way in which we could present the insights at a de-identified aggregate level to the outside community. So that is the presentation, and we can do more questions if time allows. We want to make sure to thank, as Dr. Gainor mentioned, it is a teamwork, and this doesn't include everyone, but I have here Dr. Shaw, Dr. Dagogo Jack, Dr. Hara, who's the principal investigator of the lab that we work with, Dr. Yoda, Jamie Heather, who does a lot of the T-cell work, and our pathology colleagues, and Dr. Riley as well, and all of you.
Terrific. Thank you so much for that presentation. So yeah, I guess we'd really like to hear a bit more about what you actually did in the actual 2 years of research, some of the actual experiments that you do. Is it on Petri dishes? Is it on mice? How do you do mice? And, give us a little bit of an insight into that. And most importantly, I know really what our members most want to know is, what have you found out so far, and how will it lead to the clinic? And what really everyone wants to know is, when will it lead to the clinic?
Yeah, I can start with that. So, I think this is a prime example of where, in order to tackle complex questions like this, you really need a multi-pronged approach that takes both the laboratory experiments as well as experiments based on clinical biopsies, as well as clinical outcome data. And so maybe I'll start with the lab side first, and then bring it up to the clinical side. So on the lab side, what we're really doing is we are looking for, can we find rare individuals that their immune system is already recognizing ALK. Now, let me take a step back. Why might that even be the case? I return back to that foreign concept. For your immune system to recognize a tumor, that has to be sufficiently foreign. Well, the thing about ALK is, it's rearranged. It generates this novel, this brand new fusion that the human body hasn't seen before. That's one piece, and then the second piece is that in normal development, ALK is not really expressed in the human body outside of very early on, and so it's really when you get these fusions, it leads to this suddenly new expression of ALK. And for us, that we think is a key, because it does mean that ALK could be foreign to your immune system. And based upon some studies in lymphoma, which also have different types of ALK rearrangements, there was some early evidence that patients can actually have immune cells that react specifically to ALK. And so that's one of the first things we wanted to look for. We wanted to take blood samples from patients who we see in our clinics and test, do any of those, can we find any rare patients who are already reacting against ALK? And presumably, other barriers are in the way and preventing, those immune cells from completely killing the tumor. And so, working with Dr. Heather and Dr. Hada, essentially what they're doing is they're taking immune cells from patients and mixing that with ALK in the petri dish and looking to see if there's a reaction there. And one of the most important insights that we've gained so far is we actually have found patients who actually are having reactions against ALK. And that's critical. That's a crucial first step in showing that, yes, indeed, we actually do have some, like ALK can be this, we call it a neoantigen, something that your immune system is reacting to. And what we're working on right now, this critical next steps are, one, to make sure it's specific, meaning that it's not just against ALK and maybe 50 other things, that it's just ALK and only ALK. And then the next piece is to actually figure out the blueprints, the DNA code that corresponds to that immune cell that's reacting against ALK. And those are actually experiments that we already have ongoing with collaborators from a company called Gigamune. And here, I think we've been able to shave off years, and I mean years, from the experiments by taking advantage of brand new technologies that are able to look at the DNA within individual cells, rather than where you're mixing it together. Historically, it's been a 3 to 5 year process to figure out that DNA code. Here, by using what's known as single cell technology, where you kind of use barcodes, and we can shave years off that. And so that is the critical step. And what we hope to do is, once you figure out that code, that this immune cell can react against ALK, well, then you have the blueprints, and you can create in the lab, as Dr. Lynch showed you, you can create a means by which to reintroduce that code into other people's immune cells. And that's our hope here. Is some of you may have heard about this. It's called CAR T cells or cellular therapies. These are different than the other forms of immunotherapy that you come to hear about in lung cancer. Normally, the drugs like Opdivos or Pembro, these are drugs that we're giving intravenously to try to stimulate the immune system while it's still in your body. Here, the idea would be to actually, much like you give blood, or if any of people have given platelets, essentially take out white blood cells from the body, and then using fancy tricks, insert the new DNA code into the immune cells. So now you're inserting a code that recognizes ALK into the immune cells. And the next step then would be to reintroduce those cells. Now, where are we in that process? We're at that critical stage of deconvoluting the code for those immune cells. We've already recognized that we can have immune cells recognize ALK. Now we're at the stage of, can we figure out that code, ensure it's the right code, and then that will enable us to pivot and actually create a living drug that can target ALK.
How long would you anticipate that it might take you to crack that code and have the technology to insert the code into the blood that you take from the patient?
Yeah, so we're doing all of these things in parallel to make it as quick as we possibly can, because we recognize this is crucial, and we want to jump to the clinic as soon as we possibly can. I anticipate within the next year or so, we will have those. Using these single cell technologies, we will have we hope to have cloned those T cell receptors within the next year. After that, what the next steps would be to test this in using our vast repository of all these patient-derived cell lines and animal models as a proof of principle. And then as soon as we see that proof of principle that it's safe, the goal would then to be to partner with industry in order to generate this living drug.
So for you to get to the stage where you've kind of cracked that code and got it ready for that stage, is that part of the grant that you were awarded by us, or do you require more funding for that, or how would that work?
Yeah, it's a good question. So the current grant was really focused on, can we identify these immune cells and can we start characterizing it in order to actually then validate, in animals and create a drug. This is something that Dr. Lin and I will seek additional funding from various, National Institutes of Health, the ALK Positive community. These are kind of, we think, are critical experiments in order to bring this to fruition, but it's the next step beyond this grant. So it is something that yes, we certainly need more support for, and I think it emphasizes the importance of research. And it also emphasizes that this is an entirely new way to treat things, of using a cellular therapy against ALK. It has never been tried before. And while we are, trying as much as we can to bring it to the clinic fast, since it's a brand new drug, approach, it does take, more time in the preclinical space than other therapies.
I think most of our members have heard the term CAR T therapy. Is this what you would call CAR T therapy, or is this something like CAR T therapy?
It's something like CAR T. And I glossed over the details, but the distinction is this, that for a CAR T cell can only recognize things on the surface of the cancer. Okay, it can't go inside. Okay. The challenge is that ALK when it's rearranged, is inside the cell. Okay. So how do we still attack it? And like any protein in your body, there's a recycling system within the cell. So there's a life cycle for every protein in the body. And when that happens, it gets broken down within the cell, and little pieces, little 9-segment pieces, are presented on the surface of the cell, including cancers. Now it's presented in a different way. It's kind of, we call it an MHC molecule is the technical term, but essentially it's showing, the cell is showing the world what's inside it, little glimpses of it. And so that's what we're trying to then target, those little glimpses. And so you basically need to take advantage of some of the normal machinery that immune cells use. And so the technical term here would be an engineered TCR, engineered T cell receptor. So it's not strictly a CAR T cell, but the underlying approach and the rationale is very, very similar. And so we would be able to take many of the same principles that are used for CAR T cell and apply it to this.
My understanding is that currently CAR T cell therapy is extraordinarily expensive, in the hundreds of thousands of dollars per patient. Would you agree with that short statement?
I would.
Based on that, is there anything different about the therapy that you're proposing that would be in a different, cost level to that CAR T therapy, or is it going to be kind of similar?
Yeah, so excellent questions, and I think this is not something we're necessarily producing as a we're not a pharma company, but you're right, and I think you're right to highlight the limitations of I think the drug development process, that we need to do something about the costs of drugs. This is something that I anticipate. A lot goes into treating the individual patient. For any CAR T cells or these engineered TCRs, it is quite laborious to actually develop these probes and these new receptors. So, I think for us right now, the critical piece is, can we make the biology, is it biologically possible? That's our first principle. This is unlikely, just in full disclosure. This is so CAR T cells, outside of very rare examples, those are not things that academics by themselves can necessarily create. This is something where we would then need to partner with professional cellular therapy organizations to also, again, in the spirit of these are multidisciplinary teams, and you need to go out where there is expertise, in terms of that manufacturing.
Terrific, thank you. All right, so the treatment, say it got to the point where you had it figured out and it all worked and everything, is this something that ultimately you think would actually be something to use after the ALK TKIs have failed, or would you see this as being something that would replace ALK TKIs?
Dr. Lin, what do you think?
So I think because of the success that we've seen with the ALK TKIs, at least in the initial treatment of ALK positive lung cancer, at least initially when this therapy is being explored, it will probably be in the post-TKI setting. But of course, depending on the outcome that we see, the hope would then be to explore whether bringing it even earlier in the treatment of our patients may transform their outcomes. So, most likely course is that we explore it first in patients that have progressed on all of the available TKIs. And then if we see good outcomes, which is what we hope for, then we would bring it earlier and earlier, and potentially even think about whether there's room for combining with the TKIs, there the safety and the toxicity will be the major question.
Right.
And Colin, one thing I did want to highlight, because it's different than the types of therapies we've talked about before. So in CAR T cells, patients get one infusion and that's it. You know, the drug is the immune cells which stay within the body. So essentially, these are living drugs, and so once they enter the body, they divide and they grow and then they persist. And so that's would be a fundamental difference from many of the other therapies that we're talking about here, where it'd be one infusion, and then hopefully get a response and have memory with the persistence of those cells.
Terrific, great. So yeah, I mean, coming back to what Dr. Lin was suggesting earlier about, start off in the post-TKI setting and then maybe change as time goes by. I mean, that's not a whole lot different to if you look at a new drug like alectinib or lorlatinib. When they were first introduced, they weren't given first-line approval. They were given approval only to be used after crizotinib failed, so and then lorlatinib in the case, only after alectinib and brigatinib have failed. So similar type of thing. So I understand that totally. Yeah. So my next question was, would you imagine it could actually create much longer responses than ALK inhibitors? And the answer clearly is yes. So we'll skip right over that one. Would you? And I think you answered the next one too. It could actually, the treatment can be stopped and the patient's cured, and you already, feasibly that may happen. That's the hope.
That's our hope.
Yeah.
I think that is the hope in using the immune system.
Exactly. So we had a question. For which stages of ALK positive lung cancer will immunotherapy work, or is it for all stages?
I mean, right now we still consider for early stage, cancer, stage 1, stage 2, the standard of care would be just surgery and removing it, and I don't think this would replace that. Okay. As you start getting more advanced, I think then it raises the question of how do you incorporate ALK therapies within that. I think those are the scenarios where we would aim to incorporate it.
Right. Now these are questions not related to your research, Dr. Gainor. These are more clinical type questions for Dr. Lin. There's a question here. Is there an order that TKIs are used? Can we go back to old ones? And a similar question, if someone's had progression on lorlatinib, what's the potential of going back to an older TKI?
No, that is a really good question. And I would say that not completely out of the question. There are certain instances, it's rare, I would say, but there are certain instances where if there are particular compound ALK mutations that are found, or for example, MET amplification, then we may go back to earlier generation TKIs in hopes to achieving a response. So there was the work published by Dr. Shah a few years ago, which was the first reported case of resistance to lorlatinib, where she discussed this double mutation in ALK that confers sensitivity to crizotinib. And so that patient was able to be retreated with crizotinib and have a meaningful response. So there are certain instances where recycling older TKIs may be helpful. I think in those instances, it's also important to consider patient's brain metastasis status as well, right? Because different generations of TKIs have different levels of activity in the brain. So there are multiple factors that go into that decision making, the biopsy results, the CNS disease status. But I would say we always do think about that.
Great.
I would just add that I completely agree with everything Dr. Lin just said, and I think we're coming at it from a bias. There needs to be a biological rationale to inform that recycling. Just simply going because those other drugs, were used before, we'd be less enthusiastic about that. I think it needs to be driven by an understanding of how the cancer has changed to make you think that it now can be resensitized to these.
And the only way that you can determine that's by next generation sequencing from either a liquid or a tissue biopsy, correct?
Or in
clinic.
Yeah, or a FISH test. If you're looking at MET.
Okay. Okay. So it doesn't necessarily have to be an NGS test.
We'd want that to be some component, but commonly we complement that with something else.
Right, okay. So another question here, what we as patients should expect when we get a second opinion? How should the oncologists be working together, or are we the middleman and they don't connect? When it's done perfectly, what should it look like?
So we always make a point here at Mass General, when patients are seen for second opinion, we always circle back to the primary local oncologist and make sure to convey our recommendations, what was discussed during the consultation. Patients should not feel burdened with the responsibility of writing down everything and then presenting it to their oncologist. We always make sure to do that. And then moving forward after that, it would be making sure to forward the visit notes and scan reports to each other and going from there.
I would also add that this is one situation where, COVID has caused us to all lose so much, and we recognize that. But at MGH, we've also tried to also think about how can we use this as an opportunity for our patients to also engage with us. And I think this is actually where telemedicine has actually, it's kind of kicked medicine in behind to say like, hey, catch up with the times. Mm-hmm. And, someone doesn't always have to hop on a plane to come to Boston to get that second opinion.
Right.
Right now, we are doing virtual visits just like this. And that's another medium where, I think looking at on the bright side of COVID, it's kind of forced us to actually adapt. And so that's something I would also encourage patients that also, I think thinking beyond the in-person visits, thinking about virtual visits as well. And as Dr. Lin mentioned, it's all about communication, and it's really, we view it as this is a team, and just broadening a team, right, and just continue to have good communication among everyone.
Are you using telemedicine when you're communicating with the local oncologist typically also?
Sorry, what was the question, Colin?
Are you using video conferencing when you're discussing patients' situation with the local oncologist?
I would say typically it's in the form of telephone calls or emails.
Okay.
Emails, yeah.
Okay. Do you see that might move more to video conferencing in the future, or not necessarily?
That's a really good question.
I think some. I think one way that we've tried to embrace that is, we have the MGH Core Hub, but then we have a number of satellite institutions that work with us, and I think the transition to a virtual world has enabled all of those docs to now join our tumor board, whereas formerly it was like an in-person meeting. This has actually opened it up to actually have more dialogue with all of our community partners.
So a couple more questions and then we'll wrap it up, because I know you guys have busy days ahead of you. Do you have time for just a couple more questions? So a question here, I would be interested in hearing an update on the 2 combination MEK inhibitor clinical trials going on at MGH. The one is the cobimetinib and alectinib, and the other is the binimetinib and lorlatinib. Specifically, what stage is each trial? Are there any promising results this far? And why MEK? So, you know, why have you chosen MEK as being a potential?
Sure. So the MEK pathway is known to be an important effector in the ALK signaling, and it also happens to be what we call a hub, where many of the bypass pathways that can drive resistance kind of converge. And so we have seen resistance to ALK inhibitors emerge with activation of this MEK signaling. And that can be in the form of gene mutations in that pathway, for example, mutations in MEK, BRAF, KRAS. It can also be in other forms, non-gene mutation driven ways. And we have preclinical data to support the idea of dual inhibition of ALK and MEK, where that can be a way of overcoming the resistance to ALK inhibitors. We also have preclinical data that have been published that suggests that maybe if we use that dual inhibition earlier in treating our patients, that could be a way of preventing or delaying resistance in ALK positive lung cancer. And so we're really excited about these ALK-MEK inhibitor combination trials. So the two trials that you specifically mentioned are led by our colleague, Dr. Gainor, who's fabulous. And they're both in phase 1, where we're trying to determine the optimal dose of each medication in the combination to use that's safe and that could have promising efficacy. So, you know, the primary objective is to really determine that safe dose and to look at potential side effects. But of course, we're interested in seeing any early signs of activity as well. Mm-hmm. The lorlatinib, binimetinib combination is part of a larger combination trial that was just launched. So in that trial, we have the combination with binimetinib, but also combination with crizotinib. And there will also be a combination with a SHP2 inhibitor, which we're also very excited about. And that trial just opened, and in the span of the past month, we've actually already accrued a few patients. There's been a lot of interest. And so far, we're seeing good tolerability, but it's very early. So we have to see the follow-up in the upcoming months.
And then you mentioned combining with crizotinib for the MET inhibition.
That's right.
But as we know, crizotinib is not brain penetrant, and we know that there are now some promising MET inhibitors that are brain penetrant. So you're looking to include those in a trial, or?
So that discussion is absolutely underway, where we're exploring the possibility of combining with a more brain penetrant MET inhibitor.
Okay, great. Wonderful.
Yeah, and Colin, some background there. It's a spot on question, and a question that you know we raised ourselves. I think this is where sometimes the practical also weighs in that working with different companies who make different drugs all together. That's something that can actually be a multi-year process in order to negotiate using two different drugs from two different companies. Whereas we have the advantage with the lorlatinib crizotinib combination, we're dealing with one company.
Right.
Once you actually show the proof of principle that it works, mm-hmm. then you have more negotiating ability, because you've shown that the scientific principle is right. And that, and so that's the approach that we're taking.
Okay. One last question for Dr. Lin, probably mostly. I'd like to know Dr. Lin's opinion about rechallenging with, they said Alecensa, so alectinib, or any TKI actually, after pneumonitis, if the patient has had a complete response from the TKI.
So that is a tough question. The FDA label for alectinib will say any sign of pneumonitis, because it's such a serious you should completely discontinue the drug. That said, we have had patients that have had pneumonitis on alectinib, and especially if there's been evidence of really good activity for the tumor, it's really hard to just say goodbye and abandon that line of therapy and move on. And so in certain instances, of course, there has to be a really detailed, careful discussion of the potential benefits and risks, right? Including the fact that the pneumonitis may recur, and it could recur at a less severe extent or a more severe extent. But we have in certain instances rechallenged with a lower dose with extremely careful monitoring and been successful in doing so. Now, again, I emphasize the fact that it has to be a really careful monitoring, very detailed discussion, because I don't want this to kind of become a, let's just rechallenge and move on.
Got it.
Yeah, and I would just add that it's really complicated cases like that actually Dr. Lin, I can speak for her and myself. We're always happy to jump on a Zoom and actually do a consult with a given patient and work with the local doc, just because we have a lot of experience with these things, and so we also just want to put that out there that you want to do it. But you need to be vigilant, and it really, you want to also work with people who have explored that before.
Terrific. I've just got one last question for you, and then we'll wrap it up. And I want to just again say thank you to both of you so much. What do you have left to do on your project between now and when you call it finished? The project that you're doing for ALK Positive.
Yeah, so I would say in the ordering of things. As Dr. Lin mentioned, the ENIGMA protocol will be opened. That we're really excited about. The second piece that we have we've collected all of the information now. It's just a matter of kind of writing it up. Is we've assembled the largest collections of both PD-L1 tests as well as this tumor mutation burden results now in over 300 patients, and tying that together with outcomes. What does that mean for resistance to targeted therapies? What does that mean with respect to immunotherapy? So our plan in the next few months is actually to formalize that analysis and put together a manuscript for the whole world to see. And then the last two kind of pieces are, one, completing that immune landscape using that fancy immunofluorescence. We spent a long time developing the optimal panel and validating that. So completing that analysis. And then the last piece is, like I said, we plan to try to deconvolute that code for those immune cells. And this is something that, we're going to continue even after, this is an area that we are firmly committed to. This is very much because we have a lot of confidence in this area. And so this is something we're going to continue to work on the next several years. But those are the short-term goals for this grant.
So let's work on the assumption of no further COVID shutdowns and all that sort of thing. Do you have an expected date that you would be completed with the ALK Positive study, and also an anticipated date for having an actual published paper?
Yeah, so we actually already have published several papers, from this work. Wow. The first was in Annals of Oncology that published earlier this year. A second, actually, that we submitted, we tried to turn, because we couldn't have all of our research assistants, in the labs during COVID. We actually pivoted and we said, well, what can we learn about COVID and cancer and COVID and ALK? And so we actually, as part of thinking about this and the shutdowns, we actually did just submit a paper looking at our experience, both with, how many patients had to have therapy modified, how do lung cancer patients present with COVID, when they're positive, or they present with symptoms, but it's actually their lung cancer or a side effect. So that's another paper that we've just submitted that we think is going to have some be meaningful to not just ALK positive patients, but the broader lung cancer community. We've also just submitted one paper on the looking at the mutation burden over time. So as someone moves from one ALK drug to other ALK drugs, does that mutation burden change? And working with some computational biologists, showing how that kind of the trajectory of changes. And then Dr. Lin is putting together the tying together that PD-L1 with the tumor mutation burden story. And so I think all of these things we anticipate.
Sorry, your voice just dropped out.
Sorry, I was going to say, all these things we anticipate we will complete in the next 6 months, as part of the ALK Positive grant.
Wonderful.
Yeah.
All right. Well, I know you two have busy days. I want to thank you so much. I'm glad we had an opportunity to talk without having to do it in 10 minutes.
Yeah.
And there'll be hundreds of our members watching, so even though you might feel like you weren't speaking to an audience, you most definitely were speaking to a quite large audience. And
Great.
We hope that next year we can see you in person somewhere. And
that is our hope as well.
Exactly. So
yeah, but we really appreciate the forum. Like I've said before, this is an incredibly meaningful award to us and extremely inspiring because it comes from the patient community, and we're still hard at work trying to really advance the field.
Well, I know every day you go into work, just feel like you've got several hundred eyes looking up behind you, urging you on and wishing you good luck and hoping it works out really well. Thanks very much. We'll talk soon.
Okay, great.
Thanks,
Thank you.
Thank you, Jess. Bye-bye.
Bye-bye.